The Great Barrier Reef is the largest living structure in the world and home to hundreds of different types of fish and corals. We rely on it for industries such as tourism and fishing, and it has a deep cultural value for all Australians. It's one of the best managed reefs in the world, but all reefs are under serious threat, especially from climate change, which causes coral bleaching. In 2016 and 2017, high sea temperatures caused mass coral bleaching across the Great Barrier Reef. Without a significant global reduction in greenhouse gas emissions, our reefs could face catastrophic decline by 2050. But addressing climate change alone is no longer enough. Even with significant carbon emission reductions now, we can expect ongoing mass coral bleaching and other impacts. We need to help the reef adapt to a warmer future. The Reef Restoration and Adaptation Program offers hope for its survival. It brings together the best minds in Australia and the world and will work with traditional owners, reef managers, reef industries and the wider public. It's ambitious, but the program could buy the reef precious time for survival while emissions are reduced, helping protect and restore it, and helping it adapt and survive in changing conditions. Learn more by visiting gbrrestoration.org.
Thanks, Amber. Look, I've got the pleasure to introduce the program in a few minutes and talk about its history, structure, and also a little bit about the governance. Just wanted to say that video is really summarizing our vision really well, and the fact that we working really hard to buy some time for the reef while we deal with the climate change, the causes, and obviously the impact. So the program is established as a partnership. It's a collaboration essentially of the partners you see in this slide. And it's funded by the Reef Trust Partnership and by the research partners for their co-investment. In terms of history, obviously the science that led us to this point goes way back many years. But in terms of the partnership itself, it started in 2017, where a concept feasibility study was run, leading to an investment case showing the value of R&D into these interventions and the potential benefits to the reef in the future. And that led to an endorsement by the Australian government and an investment from the Reef Trust Partnership. Since then, we've been essentially establishing the collaboration and its framework with an agreement that was signed in December last year, just on Christmas Eve. And we also have refined and developed the R&D program and the associated investment plan, which was approved for investment in August 2020 last year. So all very fresh, but since then we've been, work has started in October last year, and in the meantime, we've also been contracting and developing projects and all the details that go with it. It's a large program. We're talking of about $160 million investment combined over the 4 years. So there's obviously a lot of work in all the contracting space. Next slide, please. So the feasibility study was really taking a quite new approach here. And essentially, we looked at all the possible interventions that we could think of, or that we've heard of, or that we've been working on, and assessing those using a consistent framework based on 3 drivers, essentially, and all focused on corals, that looked at protecting the key ecological functions and economic and social values of the Great Barrier Reef, or reefs generally, be deployable at scale and feasible, as well as at a price point now or in the future that we think would allow it to be deployed at the scale we're interested in, which is entire reef scales. Next slide, please. That has led us to identifying a subset of interventions that we thought were worthwhile investigating further. And David will talk about that, but they spread across 3 different areas that talk to the prevention of exposure to temperature extremes, assisting corals to adapt to climate change, but also in some circumstances, restoring and promoting the recovery of priority reefs. And all of that assumes that we continue to intensify our efforts in what we would consider conventional management of the reefs, as well as accelerating our work in carbon mitigation and greenhouse gases reduction. Next, please. Also, during this feasibility study, we had a first attempt at assessing public sentiment, I guess, towards the concepts as well as the type of actions that we might be envisaging in the future on the reef. And generally it was quite positive, both in terms of the idea, the philosophy, as well as the need for action and the type of actions that might be considered. Of course, it's very early days, and stakeholder engagement is not only about understanding public sentiment, but also thinking of how we can work together to develop the solutions that we will consider appropriate for the reef in the future. Next slide, please. Now, the structure of the program. So essentially, the program is made of some intervention-specific sub-programs. So the conventional R&D that you would see in a specific area, to take an idea from wherever it is to a point where we think it can be applied to the reef. But also a series of cross-cutting sub-programs that really talk to the entire program and apply across the board. That includes stakeholder and traditional owner engagement, as well as regulatory frameworks. And here we're talking about obtaining social license and regulatory license in the future to use these interventions, as well as to conduct the R&D, but also to think about the participative framework that we want to employ to make sure that it's a solution that we are all owning for the reef. There's also a big component around modeling and decision support. It is a complex program, and deciding what R&D, when, where, and how to then decide to deploy these interventions, again, how much, where, is a really complex problem. And so for that, we have a dedicated modeling and decision support sub-program that is really helping us making this decision. And obviously that's underpinned by a very big effort in ecological research to continue to further our understanding of the system, feed these models with the right process information, and also make every effort that we can in understanding the risk and predicting the risk so that we can address that in the solutions we design. There's also at the bottom here, you see a program that is, and that's quite unique for an R&D program of this type. We're firmly focusing on the future and on the implementation of these interventions that we're developing. And for that reason, we're looking at how we will scale these up and deploy these technologies. So there's effort in engineering, automation, and logistics, as well as in engaging with industry and thinking about what industry might look in the future in terms of building industry capacity, employment pathways, and think about how the restoration industry might look like at various points in the future. Next slide, please. So in terms of governance, we have a board and a steering committee. It's pretty standard, just to mention that our board is made not only of the representatives of the partner organizations, but also of 4 independent members, including our chair, Rob Vitesi. Below, and all that is on the website, so please feel free to go and have a look. There's more information there. In terms of what's below the steering committee, there's a managing entity, which is led by Ames, and David is a program director. And with the program management team, they are coordinating all the work that happens across this integrated program. Next slide, please. I won't go into the detail of this, but just to say that there are a number of tasks and systems that need to be developed for the coordination and integration of this program. It's really unique in that it's very mission focused. We work as a group towards a clear set of targets and outcomes that we want to achieve. And for that, we've tried to streamline as possible and coordinate as much as possible some of the activities that happen within the program. So you have a team here that operates across project planning, field work, traditional owner engagement, all the things that you would expect in an integrated program, and communication, obviously. But below that, we have all our sub-program leaders and co-leaders, and they are driving their research teams and working in conjunction with all their colleagues to make sure that we deliver the highest quality science as possible. And you'll hear a little bit more from 3 of these sub-programs, but as mentioned by Amber, there's much more. So I'm going to pass to David now to talk about some of the scaling issues and the net benefits that we have looked at for this program. And thank you very much for the opportunity, and glad to see you all there.
Thanks, Cedric, and hi to everybody. I'm going to just briefly talk a bit about the feasibility of scale and the net benefits that a program like this may potentially deliver. Just leading into that, I just quickly want a little bit of background. So this is a graph of coral cover across the GBR, and it's based on AIM's long-term monitoring data, where they sample and analyze 200 odd reefs each year. and it has 3 lines in it. So the bottom series of red dots is the worst reef that was observed that year. The top series of dots is the best reef that was observed, and the blue in the middle is the average of all of those reefs. And there's a couple of quick observations on this. There's always reefs that are in poor condition on the Great Barrier Reef. A cyclone could have just gone through, or some other event, and so they're in a very low coral cover. There always has been, and there still are, reefs that are in pristine condition, although arguably perhaps we're not seeing as many in that condition now. It's a bit hard to tell from this data. And then the average line sort of shows this general decline, and that's the point that was, I guess, of concern to reef managers and scientists. During the concept feasibility study, we did some modelling to try and extrapolate forward to understand what might actually occur. We've had recent mass bleaching events and other things, and the reef recovers from those, or starts to recover, but again that average decline was of great concern. And this modeling kind of shows that we don't see that changing. This is looking at 2 scenarios. One's Paris, which is the blue lines, which is 1.5 degrees of climate temperature warming, and the red line is what was in those days called business as usual, or 4 degrees, and both of them showed an ongoing decline in coral cover on the Great Barrier Reef. And so raises the question, well, is there something that we can do about that? The reef is really large. Well, you all know that. It's thousands of reef, hundreds of billions of corals. The option of replacing it just doesn't exist. It's too large and too complex. So what's the actual strategy? So the first part of our strategy is that it's not one thing. It's a multi-pronged approach. Cedric talked briefly earlier that foundational is strong action on climate change, and a few of the slides I'll talk about later illustrate why that's really important. And then ongoing conventional management, water quality and cost control. But our analysis says we're going to need more than that, and so we've kind of categorized what we're looking to try and develop and make operational into 3 categories, and these are kind of listed in order of priority. The best thing you can do is protect and retain what you have, right? And Dan will talk a little bit later about some of the methods that might achieve that. Kind of next best is, can we assist the system to adapt to that climate, to that changing climate, and effectively minimize the need for ongoing intervention, i.e. do ourselves out of a job. And then when those options aren't available, then we need to be restoring those high value and damaged and degraded sites. So some of the methods are being designed to be able to do that as well. Second part of our strategy is pulling the right team together. Again, Cedric illustrated some of this by the structure of the program. And at the start of this program, we had 100 to 200 ideas from scientists from Australia and globally that we sifted through and started to assess, and we brought engineers and modelers and others in to do that assessment, and now we've formed a team that comprises all of those people. And importantly, there's a growing program of engagement and participation with traditional owners and with stakeholders in industry, because industry and others are who will help deliver this, and along with traditional owners. But of course, we're also bringing in people that are experts in financing and risk. This third part of our strategy is to, I'll just call it reuse. There's, solutions that are going to be deployable at scale are kind of this fusion between science and engineering and technology. And the good thing is that the technology largely already exists. Just by way of illustration, Lego Wars is back on the television again this week. And just a little fun fact, but Lego produce 20 billion or more pieces of Lego every year. That's 6,000 per second that they produce, they ship, they sell, and someone takes home and starts playing with them. And so the technology is not required. It's that integration with into the designs of interventions that we're actually after. And then importantly, the last point on this slide here is you need to design for the scale that's required. So if, for example, you're looking to weed a garden, you teach someone what's a weed, what's not a weed, pretty simple. And you can scale that model to a point where you can simply find more people and teach them how to weed that garden. But if you want to go to a really, really large scale, you run out of people, and it's not cost efficient. And so you would go to some sort of mechanized weeding system. And of course, the development program for those two options is very different. You don't start with a person weeding and think that, well, I'll scale that out. No, you start with, oh, I've got a large area and I need a system that's suitable for that large area. And so you might start with a small mechanized system that, as as you invest in it, you can come up with more elaborate ones that allow it to be faster and cheaper. And so that's the approach that RRAP is taking. And just by way of example, on the top left-hand corner here is a coral gardening tree. That's a great method for doing site-scale restoration. It engages stakeholders, it's simple to train people, it's visual, whole raft of criteria, but it simply doesn't scale out. You've only so many divers. The next slide there that's showing is a method that's being developed by a German company called Seacor, where they settle corals onto these little devices and then divers deploy the device. That actually allows you to jump to a next level of scale, but you can't automate it. So what's the approach? Well, you could take this type of design and split it down into its constituent parts and then allow yourself the option of bringing in kind of industrial manufacturing and processing type capability. And just as an example, during the concept feasibility assessment, we worked with some engineering companies, Worley Parsons in particular, and developed a concept for producing and deploying 30-odd million corals a year from a single aquaculture capability. And that process demonstrated that all of the engineering exists. We need to work on the biological components that feed into it. And so that's what in that program it's about. This is an example of one of these devices that we're using for testing. And in fact, this is an example of a different device from Wednesday this week, actually in the Keppels, in a project that we're collaborating with traditional owners and BHP on. And so, it's not necessarily about inventing everything. It's about taking what exists and adapting it for our use. Quickly want to talk about intervention benefits. Again, this is concept feasibility study outcomes. And as Cedric indicated, we have teams of people that are now trying to improve this and keep refining what we're assessing. This is a large deployment scenario, so 100 million corals a year, so 3 of those factories, some improved COTS control and cloud brightening being deployed. And this is now 2 scenarios. So one of these is Paris, the blue one on the left, 1.5. The other is assuming business as usual climate change going forward. And it's showing the impact of deploying this intervention in terms of coral cover. And I use this as an illustration as to why climate change is really important, right? And the more that we can mitigate, the better. Current projections are sort of 2.9 degrees, so we'll be somewhere in between this. But as you can see, same cost, markedly different benefit stream and impact. Another slightly different example, this was, if you can imagine this in the left-hand side, that's the top of the reef, the GBR, the bottom, or the right-hand side is the bottom. And in this case, a much smaller deployment, 10 million hardy corals a year, much more feasible, just on 200 selected reefs. And then the bars of whether or not over that time coral cover improved or actually got worse. And an interesting result of this assessment was that some of the reefs we were treating actually declined in coral cover compared to the control. And when we studied that, we found there was a correlation to COTS outbreaks. And so this tells us the fourth part of our strategy, that we've got to be smart about deployment. But if we are, then for the same effort, you get a much better outcome, or for the same outcome, we might only need to do a third of what this graph might have shown. And then a final slide, just quickly talking about net benefits. The reef is massive, and so you don't just assess it as the reef. We split the reef up into little sections, as shown on that diagram. And then we've been assessing different scenarios, and each of these bars is simply a different intervention strategy. It doesn't matter what they are. The top half of the screen is the benefits. The bottom half is the cost. The yellow line is the net benefit. And this shows a couple of interesting things. This net benefit, there's options that are quite economic, i.e. the benefits are more than the cost, options that are not. But a couple of quick points to note is that this was assessing those kind of simple deployment scenarios. We plan to be smart. We feel that there's at least almost probably an order of magnitude improvement that can be made as we go through this program, and understanding how to best use these new interventions as they're developed. And and made available for operational use. And secondly, this was an assessment that looked at the kind of the directly quantifiable benefits only, things that a treasurer might accept. It doesn't factor in all of those other value streams that are really, really important to the stakeholders on the Great Barrier Reef, you know, traditional owner values, or my ability to jump in a boat from Townsville here and go out and catch a coral trout on the weekend. There's a whole range of other things that come into play here, but you know, if you break it all the way back to just straight out economics, it's still a cost-effective approach. And so that's all I want to talk about. We've now got 3 talks, and I've kind of bolded them here that just give you a bit of an insight into some of the sub-programs within RRAP. So thank you.
But with that, over to you, Lena.
Well, thank you very much. And it really is my pleasure to talk to you today about enhanced corals and treatments, one of the 2 RRAP sub-programs that I have the pleasure to co-lead with Andrea Severardi. Can I have the next slide, please? And the objectives of this sub-program are to deliver key knowledge to understand the innate capacity of corals to adapt to ocean warming. And next, please. To deliver breeding methods or treatments to increase current levels of tolerance in corals. And lastly, to evaluate the benefits and the risks of these methods that I'll talk to you about in a bit more detail today. And so really what we're talking about are the corals that will be produced and delivered to the reef through the work done either by moving corals that you'll hear about, or the aquaculture program, and potentially other methods that might be applied locally. Now, we know that corals vary in their sensitivity to environmental stress, and we know that the relative temperature tolerance of corals broadly follow the temperature regimes that we see across the Great Barrier Reef. And recently, we've been successful in developing a genetic tool that can actually predict about 65% of the variation in bleaching susceptibility in individual colonies. But we still don't have a good understanding of the distribution and abundance of adaptive genetic variation across the Great Barrier Reef. And to a large extent, that's been because we've been relying on these natural heat stress events or bleaching events to show us the bleaching phenotype. Next slide, please. And so therefore, what we're now doing in RRAP is to use a standardized high throughput rapid bleaching experiment that we can deploy on the back of a boat or a research station that allows us to sample many coral species across the Great Barrier Reef and large sample sizes. And this will allow us to develop both genetic markers and maps of thermal tolerance across the reef, which is clearly of value to conventional management, but also for selected breeding. So here's the team. It's co-led by Emily Howells and myself, and we work very closely with Cynthia Reginos at UQ. Next slide, please. Now, we know that these heat-tolerant corals are able to pass their tolerance to bleaching from parents to offspring. And so therefore, if you can identify these tolerant corals, we may be able to support them becoming more abundant on cooler but warming reefs through selective breeding. And that's the second project that we're doing. And from, next slide, please, from previous breeding experiments, like the one that you can see here at a research scale, we know that the effect size can be substantial, 10 to 26-fold increase in heat tolerance when you get the genetics right. But while we've identified these potential benefits in a few species, we still don't have a good handle on how the risks identified in the literature around selective breeding apply to corals. And therefore, our work in this project, led by Kate Quigley at AIMS, will really examine these benefits and risks among more locations and species, and both in the laboratory and in the field. Next slide, please. Now, it's really beyond question that corals' algal symbionts play a central role in their health and fitness. And while these symbioses can be dynamic in some species, like you see in this image here, and respond to stress, a great number of species show great fidelity to their preferred symbiont species. Next, and previous research has shown that we can actually elevate the heat tolerance of these symbionts through artificial selection in the lab. And when we then inoculate corals with these selected types, then they can transfer these positive effects to the coral host. But it's not yet clear whether we can make the corals retain these heat-evolved symbiont strains, so even if they are of the species that corals are naturally associated with. And so therefore, Madeleine van Oppen and her team continue the artificial selection of more heat-tolerant symbionts in more species. And we use both the larval and juvenile system that we've developed already, but also an adult model that will really help speed up this research and allow us to understand the retention and impacts among life stages. And the growth, survival, and heat tolerance will be investigated both in the lab and in the field, particularly as we get a handle on the benefits and the risks of this treatment. Next slide, please. I guess one upside of the pandemic, if there are any, is that we're all much more aware of microbes and the role that they play in health and disease. And corals are really no different. They have a very complex microbial community. In ECT, we focus on the bacteria, and we work in three areas, both settlement, probiotics, and diet. Now, we weren't able to actually include heat tolerance in the current funding envelope, but we are exploring that work elsewhere. So having universal and reliable settlement queues is really a major bottleneck in any coral aquaculture venture. And we're following up on some really exciting preliminary results that show that microbes can in fact form very broad queues across a range of species for settlement. So Nicole Webster started this work, but Dr. Inke van Wondegem from University of Queensland is now taking over the leadership of this work. We're also working on probiotics for corals to help them keep healthy during the aquaculture period, as is routinely applied in other systems. And this work is led by Dr. Luna Hoy at AIMS. And lastly, we're examining the role of microbes in the diet of corals, again for aquaculture, and this is led by Associate Professor Dave Francis from Deakin and Craig Humphrey from AIMS. Now, I presented these projects as individual entities, and they are, but they are also very tightly linked. So the corals that we phenotype on the reef will indeed be used for selective breeding when we find the really tolerant ones. Likewise, the top performing algal symbionts from the selection work will also be included in the selective breeding to see how far we can push temperature tolerance and how this may or may not trade off with other important traits like growth. Obviously, this feeds into the aquaculture production, but it's also highly relevant for other coral production methods, such as the moving corals that we'll hear about shortly. We've already experienced a lot of interest from our funding partners and have secured about an additional $2 million so far for our work. ARC is pending. And we've had a very strong interest from the next generation of scientists, with over 10 high degree research students currently working in the project and several more to start shortly. And lastly, we've had the pleasure to share our plans with a large number of TO groups on the Great Barrier Reef. You can see them in the green dots. And we've discussed both our current and future engagement and collaboration with traditional owners. Now, before I finish, I would just like to acknowledge everyone involved in this program. We are indeed more than 30 people, not all full-time, working on this work. And I'd like to just acknowledge the leadership team that is helping Andrea and I keep this together. Dr. Agnes Laporte, also Rebecca Foster and Cassie Thompson, who lead our integrated field program. Thank you.
Thank you so much, Lena. We'll keep things moving and over next to Daniel.
Thanks, Amber. I'm going to talk today about the cooling and shading sub-program. And obviously it's a very large sub-program within RRAP, so I have a huge amount of collaborators across many different institutions, and the theme leaders only are mentioned there. But as you can imagine, each of those leaders also manages a whole team. So our sub-program is quickly growing to be very large. So in cooling and shading, we're interested in all of the ideas that can help to either cool or shade corals and try to prevent bleaching happening in the first place. And these ideas that we considered during the concept feasibility phase of RRAP span the full spectrum, I think, of scales. So there were ideas that apply as small as to an individual colony or a small patch of reef, right through to some ideas that could potentially be applied over the entire GBR. And of those ideas, we're only currently progressing two in cooling and shading. And these two straddle those scales. So on the left, we have fogging, and the idea behind fogging is that we would take seawater and we would mist it out. So similar to fogging devices that you might see at an outdoor concert that spray out water and the water evaporates and pulls down the air. The big difference with fogging in RRAP is that we're spraying seawater. So when the droplets evaporate, rather than, you still get the cooling effect of the air, but rather than there being nothing left, the salt crystal that was part of that seawater droplet remains. And if we target those salt crystals to be just the right size, they're very effective at scattering light, and they'll be similar to a sea fog. And this idea we can target at essentially the scale of a single reef, we think. So this idea is very well suited to protecting high value reef sites, whether they be important for tourism or traditional owner, historical and cultural reasons, or perhaps, especially as it applies to the rest of the RRAP, this might be a site where we're doing other active restoration or other interventions, and we want to get the co-benefits by relieving some of the stress on that site that we're also investing into perhaps restoring after an impact of bleaching or cyclone or something like that. On the other side of the coin, we have cloud brightening, which this idea works in a similar way in the sense that we're pumping seawater and atomizing it, but the size of the droplets is very, very different. We're creating nano-sized droplets with cloud brightening, and they quickly evaporate, leaving even smaller salt crystals. And these are targeted rather at reflecting light themselves. Those salt crystals are targeted at the size that will cause an impact on the clouds. Now, when we talk about cloud brightening, we're not talking about making clouds. What we're talking about is influencing clouds that are naturally forming so that they form as much the same cloud, but the particle distribution, the water droplet distribution within the cloud changes, and this causes them to reflect substantially more sunlight back into space. So if you look at some of our modeling results, you'll see before in the control and the cloud brightening model, the clouds look much the same, but the reflectivity of the clouds has changed. And so one of the really interesting things that came out of the RRAP concept feasibility study was that one of the very, very best things we can do for the reef is to remove the stress in the first place. And that's why cooling and shading can have such a substantial benefit. It's protecting the entire ecosystem as it is now. All of the corals and other organisms in the reef that also are subject to bleaching, such as the sea anemones. But the entire ecosystem is preserved across all species of corals at all different depths and all types. And so this is some more of the modeling that David showed earlier, but if we can do cooling and shading on a large scale over the entire GBR, for example, then on its own, it's enough under a Paris-type climate scenario to sort of halt the decline in the reef, not necessarily to head it back towards historical levels, but enough to halt the decline. As with the slides David showed you, if instead we have a business as usual climate scenario, then there's only a certain amount you can brighten the clouds. You can only get a limited amount of cooling. And so eventually that's just overcome by the pace of climate change. But one of the really important things about this slide is how well cooling and shading across all scales integrates with other interventions in RRAP. And so when you combine large-scale cooling and shading across the whole reef with other RRAP interventions, then you can see in that plot in the top right corner there, then we're able to really move the reef back towards historical levels of coral cover. And of course, these are scenarios that we're aiming to work towards, but with sort of realistic assumptions that are still to be refined. And fogging, on the other hand, we think, as I mentioned, can protect high-value reefs. And so both of these ideas, very similar as David mentioned, we're trying to adapt technologies that exist but have never been done before. So there's 30 years of research on cloud brightening, for example, but it's all theoretical. Nobody has tried to do it yet. And so we've spent the last few years very heavily on technology development, moving from the indoor laboratory testing of individual nozzles that might produce the particle sizes we need, right through towards outdoor testing, which has been taking place over the last year or two. And this is an image from the most recent field trial, which we were very lucky that the Great Barrier Reef Foundation absolutely bent over backwards to make sure to be able to facilitate this field trial while the RRAP contracting was still being undertaken. And this year, I don't know, some people might have seen the media from last year, but this year we used a sprayer that was 3 times larger in production capacity. And you can see there, this is the cloud brightening technology, and this is just the distribution of the plume coming out of that prototype, which our focus was on sampling that and how it distributed in the environment, and particularly how it was lifted vertically towards the clouds. A very interesting thing here is that with the cloud brightening, because they're nanoparticles, you can't see very many of them. What you can see here is less than 1% of the particles. And so if you imagine fogging, it would be much the same as this, but you could see all 100% of the particles. So you can imagine that would be producing a very, very dense sea fog, but we're not yet at the point of outdoor testing of fogging. And so there's just some more images from our field trial. So we were very focused in using drones this year to map that plume. And so we had many different types of drones. We had some sampling the background atmospherics, and others going back and forth, all carrying instruments through the plume. And the secondary objective of this recent field work was that there's very little background information on the atmospheric conditions, and particularly the particles in the atmosphere, the aerosols and the clouds, their interactions. And that's very important in coral bleaching and how that's going to evolve under climate change, but it hasn't really been studied up to this point. And so we have a large secondary effort in looking at those background processes that are relevant to what we want to influence to try and help cool the reef. And so, as I mentioned, it's quite a large program, and in just 5 minutes, I've only been able to cover off on some highlights, but the program has a large number of themes that go across everything you might expect for a program of this size, and essentially are a mirror image of those across all of RRAP. But we have themes on stakeholder and traditional owner engagement, the atmospheric modelling, both in terms of the efficacy of how well this will work under different atmospheric conditions and in different places on the reef and under different climate scenarios, right through to environmental modelling of what some of the risks might be of this technology if you rolled it out on different scales. I won't talk too much more about that. And just finishing up, people might be interested in the future plans, and so next year we hope to have a sprayer around 10 times of that one that we used this year, and at that scale we'll be able to start moving to using aircraft to look into the plume distribution. And then in a few more years we hope to conduct a large experiment where we'll look to measure the actual response of the clouds and to see how much brightening we actually get.
Thank you so much, Dan. And last but not least, we have the Moving Coral sub-program, where you all have the benefit of not one, but 2 of the leads joining us today to talk through their program. And no challenge, not an easy challenge for you guys, but if you could do it in about -2 minutes, that would be fabulous. And then we'll hand into questions from there.
Thanks. We'll go really quickly. Hi, I'm Peter Harrison, and I co-lead the Moving Coral sub-program with Chris. I'll talk first, and then Chris will finish off. Next, please. The Moving Corals program is a reef-based coral restoration program co-led by myself and Chris, with significant support from Matt Dunbabin from QUT. And what we aim to do is to develop very large-scale production of billions of genetically diverse coral larvae with strong environmental tolerance. And we do this by collecting samples from mass coral spawning slicks, rearing the larvae on the reefs, and then targeting their settlement onto damaged areas of the reefs to catalyse coral recovery, which underpins the reef community. Next, please. There are 4 core objectives of this project. First of all, optimising the collection of wild spawn slick samples, mass culturing many millions of coral larvae in floating reef pools, and also later in the program on vessels, and then larger scale development and monitoring of the juvenile corals that are produced through to breeding corals. In the final year, Chris will mention the plans to transfer heat-tolerant coral larvae from more northern to more southern reef systems. Next, please. Okay, so what we do in this program is take advantage of the natural coral reproduction cycles. So initially, the first phase of the project is to optimize spawn collection, and then using those eggs and sperm from these natural spawn slicks, we transfer them through to larval culturing pools and are going to optimize the development of initially many millions and hopefully in future billions of coral larvae. We then target their deployment back onto damaged parts of the reef in the third phase of the project, and then monitor their survival and growth through to reproductive size in the final phase of the project. Next slide, please. In the first phase, we're capturing coral spawn slicks. So these slicks are created on reef systems from these natural mass coral spawning events that occur over a few nights, over a few different months in late spring and early summer on the Great Barrier Reef. There are two main processes we're using. Could you return to the previous slide, please? We're using passive spawn catchers that are just anchored down current of these spawning corals, and also two types of active surface collections. Next slide, please. We then transfer the larvae into the reef pools and also do some optimization of lab rearing conditions. And we're rearing these larvae in a range of different pool sizes from 3 by 3 to 5 by 5 and up to 13 meters in the mass larval culture pools, seen on the right there. During this period, we're optimizing coral larval culture density. We're testing UV shading and heat treatments, co-culturing with microalgal symbionts, and also doing some larval staining. Next slide, please.
So one of the major challenges that we're facing with this work is how we're going to deploy these larvae onto reefs. And that's where we have a combination of approaches in our team. And that's firstly to do some hydrodynamic modeling of where high retention areas on reefs are. And if we release larvae in certain parts of that reef, well, where are they going to end up? We then could do some deployments using automated techniques, as you can see in the bottom left of the slide, and that's work being developed at QUT. And then we can also do direct deliveries from the floating aquaculture ponds, or once we've moved to the vessel phase of the operation, directly from the vessels themselves. And we can track where these larvae are going by using different techniques, including arrays of monitoring tiles, as well as using larval staining to directly diagnose where they end up. Next slide, please. And at the end of last year, we were fortunate enough to start piloting some of these techniques. As you can see in the left, we're concentrating our larvae there. And that the middle image shows us working around one of our target reefs. And in the bottom right, just starting to design some of the methods to use for direct piping of the larvae from the aquaculture pools directly onto the reef. Next slide, please. And we're also doing a little bit of investigation in collaboration with another RRAP sub-program, the CAD program. And that's to also look at differences in post-settlement survival and growth on devices developed in that program using wild coral spawn slicks that we're capturing in this sub-program. Next slide, please. And finally, so how are we going to actually do this? This sub-program is about moving corals. Now, and as Peter described in the first phase of the project, we really need to optimize all of those stages of collection and culturing and delivery. And then once we're there, we then need to be able to think about, well, how are we going to translocate corals that are more thermally tolerant to reefs that are rapidly warming using wild coral spawn slicks? And what are the best sorts of arrangements of vessels and devices to use in these scenarios? Just click that slide on, please. And that's to incorporate, to kickstart recovery on reefs, from larvae from healthy and damaged reefs. You can use those that are more thermally tolerant, and we can start to incorporate some of those heat hardening or or addition of photosymbionts to the larvae during the culturing. And next slide, please. So finally, I mean, this is a massive team project, and it's funded from the RRAP RTP funding. We have research teams across 3 institutions, 3 or 4 institutions actually, and support from research stations, and also consent from traditional owners whose sea country we've been working on. We also have a few key members who just joined the team earlier this year, Dr. Maureen Guezo, a postdoctoral researcher, and 2 PhD students who've just recently begun. Thank you very much.
So fogging is an interesting one. So the upper limit of wind speed, it's not going to be very high. So where we're hoping that fogging will help is essentially in those doldrum type conditions. So over the bleaching season in an unusually warm year, the stress in the corals gradually builds up as a result of the warmer temperatures and light. And what seems to, the hypothesis is that what sort of pushes the corals over the edge into severe bleaching is these couple of periods that typically occur each year with doldrum type conditions where there's very little cloud, very little wind, and the water really heats up a little bit extra, and also the corals are having maximum light exposure. Because there's very little wind, there's no waves on the surface of the ocean, which also help to reflect light. And so we think that the upper limit of wind, it won't be very high. It'll only be probably up to about 5 knots. Interestingly, we experienced exactly those conditions when we were out there this year, but we were testing cloud brightening rather than fogging. And so we'll know more once we get up to the point of doing those fogging field trials. But a challenge then is going to be that given that this works, let's assume for a moment that it does work and we can create the fog, the logistics of that then obviously present a real challenge. How do we make sure in an operational sense that there are vessels and systems ready to go and to fog the right place at the right time when those doldrum conditions occur? I hope that helps answer the question. Nanomet particles are not manufactured. They just result from spraying approximately 1 micron or slightly smaller seawater droplets, and then obviously seawater being 3.5% salt. When that droplet evaporates, it's the nano-sized salt crystal that's left. So we're just replicating exactly the natural process that occurs over the oceans all the time. So breaking waves create these sea salt nanocrystals all of the time, and that's what forms the cloud condensation nuclei. They're probably over the oceans, about 60 or 70% of all cloud droplets are formed around one of these nano-sized salt crystals, and the remainder are from biological compounds that escape the sea surface and also create little cloud condensation nuclei. That's something that we're looking into, but obviously it's something that's hard to know, right? So far what we've done is when looking at the large scale interventions, we've hindcasted. So we've used models to re-predict the past, those bleaching events in 2015, 2016, and 2017, and retrospectively applied the intervention. So for example, go back in time, imagine if we've done cloud brightening over that summer, how much benefit would have we derived? And so inherently, those model runs do include warm water moving onto the reef from elsewhere. And what we found was that the benefit from cloud brightening, for example, is very heterogeneous. It doesn't work as well close to the parts of the reef where water is moving on from elsewhere, because that water is not resident under the changed clouds for long enough to cool down as much as in other parts of the reef. And so that's not too surprising. But what we haven't done yet is to be able to run those models into the future. But it is part of our program. So there's some great colleagues at the CSIRO are hoping to do that, to take global ocean models that do include different climate scenarios and do sort of step through time into the future, and we'll run the scenarios there as well. So it will be incorporated. It's not yet, but it is also tricky to get it right, because predicting the future in a model is difficult, of course.
I just wanted to say that it's not we obviously been thinking about that, and I will be included in the metrics when we look at the benefits and the risk. But just want to flag that there's a space there. We're talking 5 to 10 years and potentially more for some of the big interventions and the large scale ups. That means we will look also at hydrogen and all the initiatives that are happening in this space. There are already some tourism vehicles, for example, vessels that people are looking at using hydrogen to manage their impact. So we will have to remain very adaptive to what's happening in this space because it's changing so rapidly. But we're very conscious of that in our metrics, yes.
That was a really great question because, and what I'm going to say right at the start of this is that as a group, we would not presume to make decisions about what is high value and what's not high value. You know, value is absolutely, let's call it in the eye of the beholder. There's different stakeholders, traditional owners, all value different aspects of the reef. And so it's a complex space, you know, at the very start of this, of course, we're looking at the ecological value of different locations, simply from a more mechanistic approach to, if I do something here, will I have a larger benefit than if I do something here to the broader Great Barrier Reef? Because, you know, all interventions ultimately end up as an action at a specific location at a specific time. Yeah, there's nothing aside from cloud brightening. There's none of the other interventions are kind of just sprinkling something broadly across the reef. They're highly focused. And so a big part of the program is to work with stakeholders, and we started that process and we're continuing that process to really understand how they see value, what drives value for them. And as that knowledge is gained, you know, it's those very stakeholders that are going to make decisions about deployment. It won't be this team that's making decisions about deployment. It'll be the stakeholders on the reef. Society has to agree that these interventions are going to provide a net benefit, and that you know, the risk of not doing something is larger than the risk of doing something. There's a whole range of things that come into play here, and so stakeholders and engagement with those stakeholders and traditional owners is just hugely important for this program. And so, you know, people listening in should expect ongoing and growing engagement as we come to grips with this. And there's a whole team of people in this program, the decision support area, who are really there to help try and sift through and come up with some structured approaches to then making decisions, factoring all of that kind of information as it evolves a little bit over time.
Maybe just to add to that, Amber, before we close, is to say that we're interacting very closely with the traditional owner work that is happening within the Reef Trust Partnership. And we're obviously also interacting a lot with the Great Barrier Reef Framework, whether Reef Plan or the Great Barrier Marine Park Authority. So as David said, it will be a societal discussion as well as a discussion for the custodians of the GBR and the managers as to how we progress some of these interventions. We'll provide the science.
Well, on that note, thanks again, panelists and all of our attendees. If there are any questions or comments, feedback that any of our attendees or panelists think of after this webinar, please don't hesitate to get in touch. Quick and easy way is info@barrierreef.org. And we'll make sure to get those questions and answers through to everybody. If we weren't excited about RRAP before, I'm sure we all are now. This isn't the first or the last time that you'll hear about the work that's being undertaken under this program. And we'll make sure to keep bringing sessions like this through to you, not just from the Great Barrier Reef Foundation, but from all the esteemed partners that are working together. This is very much a team effort. And make sure you stay up to date with all of the latest updates from all of the different partners. And on that note, let's wrap things up and leave everybody to their long weekends.